Okay, guys, we have here a couple of lectures, 36 and 37, about the lipoproteins, and I've divided them in two parts. One is divided as lecture 36 and the other 137. So the learning objectives of these two lectures is we have to classify different types of lipoproteins, describe briefly about their metabolism, and the relation of these lipoproteins with the cardiovascular diseases. So these lipoprotein particles are the molecules on which various lipids are transported, and these lipids can be cholesterol or any other molecules like cholesterol esters or the triacylglycerols. So these lipoprotein particles help to transport these lipids through the blood and the plasma. Surface of these lipoproteins is made up of proteins which have got a special name, apolipoproteins, and a little bit of phospholipid is there as well. So outside there is covering of lipoproteins and phospholipids. As we can see here, the blue covering is the phospholipid monolayer, and the green ones are the different types of apolipoproteins, and inside these molecules we have got cholesterol, cholesterol esters, and the DHEs, the fats which are insoluble in water, so they cannot be transported in the blood, but inside these lipoproteins they can easily be transported, and the free fatty acids, to remember, are transported when they are binded to a special protein, albumin. So free fatty acids do not transport in these lipoproteins. So there are four main types of these lipoproteins, or you can say four major classes, and their names are based on the density of these molecules in the centrifuge machine. Chylomicrons, which are the largest molecule, are the least dense. So their density is lowest, but their size is largest. And all of these lipoproteins are big enough to be seen under the electron microscope as well. Now, after chylomicrons, we have VLDL, which will have a little bit smaller size than chylomicrons, but their density will be a little bit higher. Then comes low density lipoprotein, and last one is the high density lipoproteins, which are the smallest in size, but their density is the greatest. So a few questions can be there regarding these different types of lipoproteins. What you have to remember is which one is the largest, which one is the smallest, which one has got the highest density, which one has got the lowest density. So chylomicron density is the uh, lowest, you can say, and their size is the highest. And another point to remember about chylomicrons is that the triacylglycerols or THEs are the greatest in these chylomicrons. Then we have the HDL, which we just talked about, smallest in size, but their density is the greatest. And these HDL have got the highest amount of proteins in them. So such a question can be there, which one of the following has got the highest amount of protein in them? Those will be HDL. Which one has highest amount of THEs? That will be the chylomicrons. So we have the electron microscopic pictures of these molecules, and we can see chylomicrons, the biggest ones, and HDL are the smallest of these particles under the electron microscope. So the main function of these four types of lipoproteins are mentioned on this slide. Chylomicrons, sometimes shortened as the CM, and they carry the TGs, which is triacylglycerols, from intestine to the liver. So absolutely important to remember their function. They transport TGs from intestine to the liver. VLDL, or very low density lipoproteins, also transport THGs mainly, so they carry TGs but from liver to other body cells. So that is the difference between the two. Both mainly transport triacylglycerols, but and the difference is from which organ to which organ. Then LDL, the main transport is cholesterol, and it transports to all the body cells. And high density lipoprotein, which is considered to be a good fat from the body, uh, brings the cholesterol back from the body cells to the liver. And this is known as reverse cholesterol transport for the HDL. So this slide is about the different types of the apolipoproteins. Uh, those are the proteins that cover the lipoprotein particles. So these are of different types and are found in different types of the lipoproteins that we just saw in the previous slide. Some of the important ones in these are ApoC2 and ApoC3. ApoC2 activates an important enzyme lipoprotein lipase, whereas ApoC3 inhibits the same enzyme lipoprotein lipase. Similarly, ApoB48 is also important, which is involved in cholesterol transport and its clearance, whereas the ApoB100 binds to the LDL receptors. So these are a few noteworthy ones. ApoE is involved in the clearance of VLDL particles and the chylomicron remnants, whereas ApoA1 and A2 again activates and inactivates an enzyme ALCAT. So a brief description of the chylomicrons and the VLDL, and how is the lipid content inside these lipoproteins is activated and metabolized. So we just saw in the previous slide the protein ApoC2 is the one that activates the enzyme lipoprotein lipase in chylomicrons, and this enzyme breaks down the TAGs into free fatty acids. So we get the fatty acids which will be released and can be used for energy or fuel in many different tissues, adipose tissue, heart, and skeletal muscles as well. When the fats are depleted, the remnants will go to the liver for absorption. So once all the energy has been extracted, the leftover parts will go to the liver. Similarly, in VLDL, again the same protein ApoC2 will activate the same enzyme lipoprotein lipase and releases the free fatty acids. And adipocytes or adipose cells will take up these fatty acids, reconvert them back to THEs, and store them in the lipid droplets. So the function is a little bit different here because they are going to be stored in the lipid droplets, whereas in the muscles these TAGs can be used for energy purposes. So when it comes down to the low density lipoprotein or the LDL molecules, the muscle and adipose tissues have these LDL receptors which will recognize the protein ApoB100. So this apolipoprotein is important regarding the LDL ApoB100, and that is what is recognized by the receptors. So muscles and adipocytes will take up the cholesterol from these LDL molecules, whereas HDL, the high density lipoprotein, is considered to be a good lipoprotein or a good fat in the body because it picks up cholesterols from different parts of the cells and return them to the liver, where we can metabolize these cholesterol, convert them into bile salts or bile acids, or throw it out of the body. So this HDL helps us in lowering the cholesterol levels in the body by this reverse cholesterol transport, and the remnants of the cholesterol will be converted to cholesterol esters by it as well. Next up, we have the metabolism of chylomicrons, and the statement at the top is the most important one, that chylomicrons transport exogenous lipids. The lipids that we eat in our diet that come from outside the body are known as exogenous lipids. So these chylomicrons help in their transport. So what happens to these exogenous lipids once we eat them and they are broken down into small particles? Then in the small intestine, With those lipids, we make this nascent chylomicron particle, which can be called as a baby chylomicron because it's a small molecule. So from the small intestine, it goes out and it has to go into the bloodstream, and on its way it takes up two lipoproteins, ApoC2 and ApoE from HDL. So HDL donates these two proteins to this chylomicron. After taking up these two lipoproteins, it becomes a mature molecule known as the full chylomicron, or we call it a mature chylomicron as well. It had one ApoB48 of its own, and it has taken these two C2 and E from the HDL. So now it has got three apolipoproteins on its surface, and inside it has got lipids, the greatest quantity of this TAG, then cholesterol esters, which is shortened as CE, and the C stands for cholesterol, which is in the minimal amount. So this chylomicron goes into the bloodstream, which is represented by this capillary, and inside the bloodstream we have this lipoprotein lipase enzyme, which is activated by this ApoC2. So ApoC2 activates it, and this is also a good point to remember, that this protein activates this enzyme, which is lipoprotein lipase. Now this lipoprotein lipase breaks the TAGs in these chylomicrons, which was in the greatest quantity. The TAG will be broken down into two parts, free fatty acids and to the glycerol molecule. So most of the TAG is broken down. Fatty acids are taken up by different tissues like adipose tissue or skeletal muscle, and it will be used by those tissues. Glycerol will go back to the liver and can take part in glycolysis. Now the rest of this chylomicron comes out of the bloodstream, and it will return one of the proteins, ApoC2, back to the HDL. So it goes back, and what is left is known as a chylomicron remnant. Now the things to remember is it has got two proteins on its surface, ApoE and ApoB48. The third one, it returned it to the HDL molecule, and inside the chylomicron now TAG is in the smallest quantity. In the full mature chylomicron, it's in the largest quantity, but once it is broken down in the blood here in the capillary, now TAG is in low quantity and cholesterol ester is in high quantity, and this molecule is known as chylomicron remnant. It will be taken up by the liver. Liver has a receptor which binds to the ApoE, and so a receptor is on the surface of this liver cell. We can see here this is the receptor, and the ApoE recognizes it and binds with it, and the liver cell takes this chylomicron remnant inside. The process is called endocytosis. Second is we have got the VLDL metabolism, and also in this photo is also the LDL metabolism, which is pretty similar to the previous diagram that we saw for chylomicron metabolism. Important thing to remember here is again at the top, VLDL transports endogenous lipids. So this one transports endogenous lipids, the lipids that are made inside our body. Those are the ones we called endogenous, and the ones that we eat in our diet were exogenous, as you saw in the previous slide. So in the liver is this VLDL molecule going to be prepared. It has got one protein on its surface, ApoB100, and it goes out of the liver. Again, like chylomicron, the one that is made earliest is known as nascent VLDL, or you can say baby VLDL. And it goes outside the liver cells, and before going to the blood, it takes two proteins from the HDL, ApoE and ApoC2, and matures up, gets bigger in size, and we call it the full VLDL molecule. Now has got three proteins outside, the B100, ApoE and ApoC2. And again the composition is the same, most or highest in TAGs, then cholesterol esters and cholesterol in low quantity. Then it goes into the bloodstream like capillaries, and the same thing again happens. The protein ApoC2 will activate the lipoprotein lipase, same enzyme inside the blood, and lipoprotein lipase will break down the THE molecules into the same parts, free fatty acids and the glycerol. Same fate for these two. Free fatty acids goes to different tissues like adipose or skeletal muscles and can be used there. Glycerol goes to liver and is used mostly in glycolysis. After the breakdown of THEs, this molecule comes out of the blood and is known as IDL or intermediate density lipoprotein. Then it gives away two proteins. So the two that it took from HDL, it gives both of them back this time. So a little bit difference from the chylomicron, in which it only returned one. Here it returns both of them, and the remaining particle is what we call the LDL or low density lipoprotein. It has only got one protein on its surface, ApoB100, and TAG again is in the lowest quantity. In VLDL it's in high quantity, in LDL it's in low quantity. Cholesterol and cholesterol ester are in high quantity. This LDL can be taken up by the extrahepatic tissue, which in this diagram it is represented by the muscle cell, or it can go back to the liver. Both of the or any of these organs which take it will have the receptors, and the receptor will detect this ApoB100 protein. It will match with it, and then it will be endocytosed or taken inside the cells of these tissue. Liver or any extrahepatic tissue, most commonly is the skeletal muscle. So we saw in the previous slide that LDL carries the cholesterol from the liver to different organs, primarily the muscles and the adipose tissue. So we look at the endocytosis process in a bit of detail, how this LDL particle is taken up by this muscle cell or the adipose cell. For taking these LDL molecules inside on the surface of this LDL, we have got the protein which is ApoB100, and this protein is important because this is going to recognize the receptors which are shown by these green molecules on the surface of the cell. These are called the LDL receptors. So this green molecules, LDL receptors, they bind with the ApoB100, as we can see here. That receptor and the B100 protein binds, and then it is taken inside of the cell. Once it is inside, it is coated vesicle coated with the clefrin pit, and once it is completely inside, it will be taken up by the lysosomes, and the lysosomes have the enzymes to break the particles that are inside this LDL, whereas these green receptors will recycle back to the surface. As we can see here, they are going back and they will attach on the cell membrane again, where they can pick up more LDL particles. So the contents of this one, which was inside the LDL, they will be broken down by lysosomal enzymes. So it will break down into amino acids, if it has any, mainly fatty acids, and the cholesterol, which is the most you can say important component of this LDL. So cholesterol is here, which was broken down from the LDL particles. Some cholesterol is synthesized in the cells as well, so synthesis of cholesterol can also contribute to this cholesterol. And cholesterol has got many functions to play in the cell and in the body. We can use it for cell membranes, steroid hormones, and bile acids. So these are three main functions of the cholesterol. And if there is extra cholesterol in the body, we can use the ACAT enzyme and store cholesterol in the form of this cholesterol esters, which attaches a fatty acid with it. When there is too much of cholesterol, there will be oversupply of cholesterol, so we have to lower the levels of cholesterol in the body. So oversupply or high levels of cholesterol inhibits the HMG-CoA reductase enzyme, so we will stop synthesizing cholesterol. Also, high cholesterol is going to act on the or inhibit the synthesis of the LDL receptor, So here is from the DNA to messenger RNA, and on the endoplasmic reticulum, the receptors are being synthesized. So high cholesterol will stop this synthesis, as we can see the negative sign here. So this synthesis will be stopped, which normally is done in the endoplasmic reticulum, and then these green LDL receptors goes to the surface of the cell membrane, so they can take the more and more LDL inside. So this is the whole mechanism by which LDL is endocytosed and is broken down in these different cells, chiefly muscle cells and the adipose. The main thing to remember is that the LDL receptors bind or recognize the ApoB100 protein, and then it is taken up, and this whole process is known as receptor mediated endocytosis. So this one is simply the description, you can say, of the diagram which I just explained in the previous slide, the receptor mediated endocytosis. So the LDL receptors, which are negatively charged, they are present on the protein calithrin on the cell membranes, which stabilizes the shape of the pit. Then these LDL receptors bind with the ApoB100 protein, and it is internalized. That is the process we called endocytosis. And the vesicle loses the calithrin coat, and it will form large vesicles known as endosomes. The pH of the endosome falls, and the low pH causes LDL separation from its receptor. The receptor goes back to the cell membrane, as we saw in the diagram. So receptors can be recycled back, whereas the lipoprotein remnant was broken down by the lysosomal enzymes to its components, and we saw some amino acids, some cholesterol, etc., that was formed from the breakdown of LDL. Then is the last part, as I explained already. When we have excess cholesterol, means high levels of cholesterol, what will its effect be? It inhibits this HMG CoA reductase enzyme, as I just told you, to stop the synthesis of cholesterol. It will repress the LDL receptor protein gene expression, so we will not make too much of the receptor. So receptor synthesis will be stopped basically. So this limits the LDL cholesterol entry into the cell, so that we will not be taking more LDL inside the cell, because there won't be many receptors present. And another function of this high cholesterol is, which I forgot to mention, is it activates the ACAT enzyme. And this enzyme, which I told you, that is going to synthesize cholesterol esters from the cholesterol molecule. So when we have too much cholesterol, obviously we can store it. So in this scenario, high cholesterols will activate this ACAT enzyme.